Hello, my name is Merran Hague, and I am a stock assessment biologist with the Pacific Salmon Commission. My presentation discusses the challenges surrounding the in-season stock assessment of Fraser River pink salmon. In this presentation, I will cover the role of the Pacific Salmon Commission, provide an introduction to Fraser River pink salmon, describe our key stock assessment tools used for in-season assessment, provide a description of recent trends in pink salmon behavior, biology, and run size, and discuss the potential for using environmental covariates to improve or supplement our current suite of forecasting tools. The Pacific Salmon Commission is a bilateral regional fisheries commission formed to support the implementation of the Pacific Salmon Treaty signed between the United States and Canada in 1985. The staff of the PSC Secretariat provide administrative and biological support to help the two parties meet their responsibilities under the treaty. We are actually one of two fisheries secretariats located in Vancouver, British Columbia, Canada, and the NPAFC staff are our close neighbors. The PSC is comprised of a series of panels and technical committees. As a staff biologist, my role is to provide support to the Fraser River Panel, a multi-stakeholder group responsible for the management of fisheries targeting returns of sockeye and pink salmon to the Fraser River, British Columbia. Together with my colleagues, we administer and operate marine and river test fisheries, collect biological samples, develop, run, and analyze stock assessment models, and provide near-continuous reporting of updated assessments to the Fraser River Panel during the fishing season to help inform their management decisions. The Fraser River system is unique in its data richness, and in-season management is a complicated and impressive system of sample collection, genetic stock identification, scale-based aging, test fishery, and hydroacoustic abundance estimation, with near daily turnaround of updated stock assessments. The Fraser River is Canada's largest river discharging into the Pacific Ocean and is home to British Columbia's largest run of pink salmon. Pink salmon migrate directly into the ocean as fry and return to the Fraser on an odd-year cycle. Commercial, recreational, and First Nations fisheries harvest pink salmon returning through marine approaches along the inside or northern coast of Vancouver Island and the outside or southern coast. In addition, fisheries occur within the Fraser River itself. Historically, annual returns of pink salmon were often in the tens of millions, but returns have been declining in recent years, and the pre-season forecast for 2021 provided by Fisheries and Oceans Canada is for a record low 3 million return. In season, the PSC Secretariat updates estimates of return timing and run size using information from marine test fisheries located along both approach routes, eventually updating with more accurate estimates using data from a hydroacoustics facility located in the lower Fraser River. Despite these multiple monitoring locations, providing accurate in-season updates to returns of pink salmon continues to be a challenge. My presentation will focus on some of the tools we use, what's contributed to these challenges, and some of the approaches we're hoping to use moving forward. While we have fairly robust approaches for updating our estimates of sockeye salmon return to the Fraser River, pink salmon in-season stock assessments continue to be a source of uncertainty. To try and improve the robustness of our estimates, we use a suite of tools for assessing the size and arrival timing of the return using our marine test fisheries located seaward of key fishing areas. These methods include daily estimates of test fishery catch unit effort data converted into estimates of abundance using historical estimates of catchability, applying Bayesian normal time density models fit to test fishing CPUE data and also informed by historical and forecasted priors, and finally, run reconstruction models, which we use to align abundance estimates and fishery data from alternative locations to compare and combine sources of information. Historically, we also relied on other simple linear regression models such as run size versus fish weight or run size versus marine arrival timing. But these relationships have either changed direction or broken down completely in recent years. This has reduced our suite of available tools and led us to question what's driving those changes. In the following slides, I'll review some of these tools and discuss their underlying assumptions that contribute to some of the uncertainty in our assessments. The first and most simple approach is a relationship between test fishery CPUE data and the size of the run. Using a 3-day average CPUE collected 10 days before the peak of the run migration provides an early indication of run size. While it can give an indication of whether the run is likely to be larger or smaller than the preseason forecast, it is more informative for smaller run sizes as there's evidence of density dependence in the catchability relationship. These models require some knowledge of the timing of the run as the relationship weakens later in the season and have an underlying assumption of consistent or equal catchability both across and within years. We also have a more sophisticated tool for predicting abundance, as well as marine timing from our test fishing data. Details of this model can be found in a 2019 publication by PSC staff. Assuming a normal entry distribution, we use a Bayesian run timing density model to fit a normal curve through our abundance estimates derived from marine test fishing data. Priors for the mean standard deviation and magnitude of the normal curve are based on historical data or preseason forecasts of the timing, run size, and spread, where spread is the number of days encompassing 95% of the return. CPUE is converted to abundance estimates based on a historical distribution of catchabilities. The fit of the model is continually updated as more days of CPUE data become available. Again, precision and accuracy is affected, one, by the limitations of the model, as the shape of the return is typically multimodal or even skewed. Given uncertainty in the data, the prior can also be influential and can bias results. And finally, we assume that the catchability in the current year is similar to historical observations. This tool is typically most informative when used close to or just following the observed peak or median of the return. Our third tool for estimating pink salmon is a backwards run reconstruction model. Starting from a known escapement estimate, we work backwards to incorporate seaward catches and recreate daily abundances of salmon at marine entry. While a relatively accurate approach in the postseason, once populated with observed estimates of migration rate and postseason escapement, we also strive to use reconstructions as an in-season tool for combining multiple sources of relevant data as pinks migrate through marine approach fisheries. This includes data from our test fisheries, catch and catch per unit effort data from commercial and indigenous fisheries, and hydroacoustic estimates from the river. The two key underlying assumptions of these models are that fish maintain order of movement like a series of train cars, and that there's a minimal and consistent delay for fish traveling between marine test fisheries and downstream areas, so that we can still provide timely updates to inform fisheries openings or closures or calculations of total allowable catch. Ideally, we would want to incorporate information from our hydroacoustics program in season, but unfortunately, pink salmon migration behavior between marine and freshwater areas fails these two requirements, as fish often show changes in distribution and one or more periods of delay before reaching the river. An example of the potential benefit of the reconstruction model and the usefulness of commercial catch per unit effort data is illustrated by this example from 2013. Our test fishery, shown in red, was suggesting record high numbers of pink salmon migrating around the southern end of Vancouver Island. However, the CPUE data provided by the US commercial fisheries suggested a much smaller run, which was ultimately confirmed using postseason hydroacoustic estimates. The US data has higher and less variable catchability and a larger sample size, or more vessels, which means it can potentially provide a better run size estimate. However, if there are few fishery openings, our reconstructions are more susceptible to failure of model assumptions regarding migration timing or offset. For example, if we focus in around this area on September 1st, we may have resulted in the opposite conclusion, that the test fishery was underestimating run size compared to the US data. Precisely this kind of misinterpretation occurred in 2019, when the salmon displayed their earliest marine arrival timing and longest travel time between marine approach areas in the river than we had ever observed. As a result, incorrect assumptions were drawn when aligning assessments from multiple locations. As I previously mentioned, some of our pink salmon time series have changed in recent years. Many of these changes appear to have started in the late 1990s or early 2000s, aligning with regime shifts in other salmon assessments for the Fraser River, including changes in sockeye salmon productivity. This was especially noticeable in 2019, when fish arrived exceptionally early and migrated slowly between marine areas and the river. Our stock assessment models are prioritized or rely on priors based on historical assumptions. As a result, the early timing, slow migration rate, and narrow distribution resulted in biased and uncertain estimates from our normal suite of assessment tools. Now that we have sufficient information to characterize the non-stationarity and variability in some of these trends, we wanted to explore underlying environmental conditions to see if they could help explain some of these differences in pink salmon behavior as well as run size. Including environmental covariates in Pacific salmon stock recruit models is becoming more common. For example, this year, Fisheries and Oceans Canada biologists used different environmental covariates to predict returns of pink salmon to the Fraser River expected later this summer. In a series of retrospective analyses, we observed that covariates such as the Pacific Decadal Oscillation, spring or summer sea surface temperatures, and river discharge conditions were useful predictors for both pink salmon run size and arrival timing. Regional sea surface temperatures from the ocean entry year in the spring are negatively correlated with Fraser River pink salmon run size. Temperatures from the same location, but during the return year in the summer, are also correlated to arrival timing and marine test fisheries. These relationships were not previously significant or useful for forecasting, but they have strengthened in recent years. This is due to the increased frequency and magnitude of the environmental anomalies, as these relationships are usually strong when the anomalies are more extreme. Pink salmon behavior has been changing, and our stock assessment tools need to adapt. Updated model parameterization and use of environmental data to predict annual timing and abundance of returns may help improve our in-season assessments. Models should be iteratively reevaluated, particularly until causational relationships between environmental data and biological responses are better understood. The 2021 pink salmon run experienced record high temperatures upon marine entry, and we will know in a few months whether the corresponding prediction of low run size materializes. Future research will continue to search for environmental covariates that can help inform our in-season stock assessments. With thanks to collaborations from PSC Secretariat staff, the Fraser River Technical Committee and panel, partial funding was also provided by the Pacific Salmon Commission Southern Endowment Funds. And finally, thank you to the North Pacific Anadromous Fish Commission and conference organizers for this opportunity.